And what I want to draw your attention is this picture, little arrow A and B, and you can zoom into this little arrow A and B. And what you see here are loads of little spiny things. It looks a bit like, like a Christmas tree in high resolution, but actually what it is, it's synapses. It's connections from other neurons that make connections to this neuron. And of course, a cell is never sitting on its own, and a recently developed imaging technique allows us to see all the cells simultaneously. By some magic of genetics, they can basically stain each neuron in a different color, and all its parts are also stained in different color. So what you see here is the more realistic picture of your brain. You see individual pyramidal cell, like here, but what you see is there's a dense feltwork of connections. And that dense feltwork is your connectivity. And just to get a feeling for how many of these little cables there are, one neuron makes roughly 4 centimeters of these connections. And this means per cubic millimeter, so that's the volume under a very well-trimmed fingernail, you have 4 kilometers of these little cables whizzing around. Okay? That's very dense connectivity. So let me show you another picture of neurons. It's over 100 years old, drawn by the anatomist Santiago Ramon y Cajal, the first Nobel Prize winner in medicine, and he invented words like synapse and neuron and so forth. And all of these are brain cells that you find in your brain. This is the pyramidal cell that I showed you earlier, and there are a whole set of other neurons. And you can see how vastly different. There's a whole zoo of them, but all of them have basically these structures, these trees, these dendritic arbors we call them, that try to collect information, and then collect this information, process something in the cell body, and send out the information to another cell. So this is the stuff that makes your brain tick. And instead of going more into the details of that, let's take a whole step back. Let's zoom out of the brain, and let's look at something what these little neurons do. So many of you may have seen this picture, but some of you may have not seen this picture. There's an animal in this picture. Who can spot the animal? Okay. Who cannot spot the animal? Okay. I'm going to show you the animal. And I just need to show it to you once, and for the rest of your life, you will spot the animal in this picture. So if you don't want to be marked for the rest of your life, close your eyes. Okay? There it is. It's a dog, it's a Dalmatian. Now, if you're a computer scientist, you're quite frazzled by that, because how on earth are you connecting these black blobs together to think this is the leg of a dog? Yet, whenever you will see this picture again, you'll see this is the leg of the dog, this is its back, as if there were lines, but there are clearly no lines up here. There's just, black and white patches. But you're painting and filling in what's actually there. So this is really amazing stuff. Let me show you another visual illusion. So in here, you see a two-dimensional picture. It's projected on the wall. And you see stuff sticking in and stuff sticking out of this picture. And now I can ask you, how many of you think that this guy sticks in? Hands up. Okay? How many of you think this guy sticks out? Hands up. Okay. And so you would think this also sticks out, yeah? Okay. So let me just rotate the picture a bit. Same picture. Who just changed their mind looking at the same picture? So how can we explain that? Well, it explains that the process of perception is more than just pure sensation. It's more than just pixels in our retina or pixels that you take up in your camera. There's processing of information going on. And what you actually do here is you're aware of the fact that usually light comes from above. And hence, if there's a highlight on this side here, it means something sticks out, and the shadow above, and this must be also sticking in there. So you're combining knowledge about the world with your perception, with your sensation, to produce perception. So your perception is a construction. You don't want to tell that a lawyer or a judge too much, because there are some implications for law here that we don't want to exploit. But what it does mean is that several of the perceptual illusions I'm going to show you arise from the brain operating with a rational system that is designed to make judgments in the face of uncertainty. And you face uncertainty here because this picture is ambiguous whether this sticks in or out. But you have to resolve this ambiguity to be able to make decisions. So you fill it in with what you think works. Let me show you another thing, and this is really mean to all computer scientists present here, should there be any, such as myself. Imagine you have a visitor from an alien planet, and he brings all these nice goods here. These are all computer-generated fractals. They're not real objects. They're generated. You've probably never seen them before in this way. And I'm pointing out to you now that the alien recognized that this guy here pointed at it and said, this is a tufa. Now, can you in the audience point me to other tufas on this picture? Who spots at least one other tufa? Who spots at least two tufas? Okay. Let me ask you a bit more. Do you think this is a tufa? Do you think this is a tufa? Is this a tufa? Okay. So now let me ask you this thing. If you were to come in a class, basic mathematics, high school, and the teacher shows you an equation, and he tells you, this is the result of that equation, would you have understood mathematics out of this single equation? And be able to write down, multiplication and addition rules and so forth? No. But yet, I show you a single example of an object from an alien planet, computer-generated, completely artificial. And yet you can basically come up with a whole anatomy and tell me that these are tufas, but this is not a tufa. I just showed you a single example. Yet you're painting in and filling in all the rest. So this is something we would like to be able to do as engineers, make our computers so smart that from a single example and reasonable assumptions about the world, we can make deductions about how all of this happens. So this is really a challenge. So what we can then do is continue this experiment and show you other examples of tufa, and then observe how your beliefs about what tufas are change. So at some point I could tell you, well guys, this is also a tufa. All of a sudden, if I tell you, this is a tufa and this is also a tufa, your whole belief system about what are tufas changes instantaneously. And I can ask you questions and you can answer these questions as if nothing happened. This is amazing. For a computer scientist, this is really something I would like to be able to achieve. Okay. So, but before I go off onto computer science, which I don't want to do tonight, I want you to make a bit more appreciate what you actually have sitting up in your head at the moment. So we can make a comparison between your cerebral cortex, this thin tissue of 2 millimeter height and roughly 18 square feet, is the cerebral cortex, and it contains something like 2.5 billion neurons. And each of these 2.5 billion neurons has something like 10,000 of the little spines, these so-called synapses. Now we can take a fairly fancy, lightweight, energy-saving laptop like this here by Apple, and this chip that I have there has roughly 291 million transistors. These are the little things that switch on and off. And if you're really mean to the biology, we can say one transistor is like a synapse. And that's completely unfair to the synapse. But let's just stick to it for the moment. Let's carry on with our comparison. So your cortex weighs roughly 1.3 to 1.4 grams. The CPU is lighter, 370 grams. It has a lot more volume than your CPU. Clearly there are blood vessels and stuff to keep it alive. But here we're hitting one interesting thing. It just consumes 16 watts of power. You couldn't read with a light bulb of that strength. While the CPU here consumes 34 watts of power. The signal that it uses, the impulses in your brain, are about 100 millivolts, or a tenth of a volt high. And these guys use something that's 10-fold bigger. And usually a neuron fires off a signal once every second, or 20, up to 20 every second, but not more. While this guy hums at something like 2.3 billion impulses a second. Now comes the interesting thing. If we look at the wire sizes of these circuits here, they're somewhere between 0.1 micrometer to 20 micrometer. That's in millionth of a meter, much, much thinner than any hair. And actually CPUs are roughly in the same ballpark, 0.045 to 0.1 micrometer. Roughly the same ballpark. But what I want you to make available now is a direct comparison between the brain and the CPU. And that's this. the number of computing elements, so we're equating a synapse to a transistor, is roughly 800 times higher per unit volume in your brain than in this fancy laptop. It weighs roughly 1,500 times less per computational unit than that laptop. But what's really mind-blowing is each computational element in my head consumes 200,000 times less power than each transistor in that laptop. Now, what does this mean? To make that really appreciable, it means if I charge my laptop or my mobile phone today and let it compute something, it can compute for 450 years on the same charge non-stop. That's how energy efficient your brain is. So your great-great-great-great-great-grandchildren can still operate the device with the same charge if we will be able to unlock how the brain becomes so energy efficient. Now let's look at what's going on inside your brain. So I'm going to see you classic data that won some people a Nobel Prize roughly 40 years ago. And basically what they did is they showed a moving dot on a screen to, in this case, a monkey, and the dot moved. Light dot was observed by the monkey, so as the dot moves, it traced a path over the retina of the eye. And each of this, in the retina, you have loads of little cells, so-called ganglion cells, so these are neurons that look more like star-shaped, basically. And as the dot moves through, it basically stimulates this neuron. And what I'm going to make you hear is this firing of the action potentials, these little brain signals. Oops. This is the sound your brain makes 2.5 billion times across the whole brain. And what they're basically doing is they're basically moving the light dot to work out where exactly does the neuron that we're looking at observe the brain. And it doesn't respond to darkness. It doesn't respond to darkness around that region. Clearly no response, but if we move the dot through the center, like there, it responds. Okay, so this is a single brain cell responding to input from the brain. So the past 30 years since then, so roughly till 10 years ago, much of the research was focused on understanding what was going on in a single brain cell, because it's such a complex beast. But I want to give you a reason why that's not enough. Looking at the individual elements is not good enough. So consider this. Look at this little block here that I have. And let's do the same experiment that we did just with this brain cell in your mind in this room. So you're a brain cell, so you're looking at this little round region here. And I'm going to start to move that little block. Now with your hand, can you show me the type of motion you think that the block does? Okay, this is what most of you do. Okay? So now let me just take away make you aware of what's going on around you. Okay? So this is what we call the aperture problem. If you look at a small thing of the world, things become highly ambiguous, and you cannot distinguish things. And that's why it's not a smart idea to look just at single cells. Let me show you another example here. So this is another classic example. Look at this square A here and this square B here. Who of you thinks that A is darker than B? Who of you thinks that B is darker than A? And who of you thinks that both equally dark? Okay? So again, let's limit our view of the world to that of the cell. And in this case, you will see that A and B are equally dark. In this case, the other cells in the brain were telling these neurons that we're looking here, well, look, there's a shadow falling here, right? So the shadow clearly must make things darker than where things are not under light. So therefore, you must assume that the brightness here is higher than there. And again, it's an example of how we can use visual illusions to reconstruct something about the world and how your brain processes it. So now I've convinced you that it's not good enough to look at single cells. We have to look at these cells working together to understand what's going on. And now making a bit of a jump, because instead of trying to understand how these things calculate simple things like visual images, we want to understand something a bit more medically relevant. We want to look at brain-machine interfaces. So how can you, these little cells that you have in your brain, control your arm, be it your real arm, or if you've lost your arm, a prosthetic arm. And so this was some classic work done in the past 10 years. So what you see here is a monkey. The monkey is offered food, and it's using a robotic arm to feed itself. And what basically my colleagues did is they put some electrodes into the brain of the monkey, and the monkey is fully okay. The monkey still has an arm, and observed what the monkey was doing while it was eating. And from that worked out what the brain cells that they were looking at were doing when the arm was in a special position. And then basically they hooked up a robotic arm, basically bound the real arm behind the back, and whenever they were seeing the same pattern of activity in the brain, they were moving the arm accordingly. Okay? That's the idea of brain-machine interfaces. But what then happened is actually, before they could make this video, the monkey had roughly to try for roughly 6 to 9 months every day for 8 hours to feed itself with this robotic arm. And that's because we're not very good at reading out what's going on inside the brain, because the brain is highly variable. But this is state-of-the-art, invasive, because we implant something into brain-machine interfaces. And this is what I'm really going to talk about today. But let me start with a simple question. Why do you think we have brains? Since we're talking about them, I think that's a very legitimate question. Any ideas? So, sorry? Survival. Okay, that's cool. Something more specific. Yeah, great, yeah. Vision processing, okay. Any other ideas what we need? Workouts was first. It's all sensory. Okay, so it's great. It's exactly what I wanted to hear. And now let me draw you attention to this picture. And so all these guys over here have brains. All these guys over here do not have brains. All these guys over here are plants or fungi, and they just stick where they are for the rest of their life. All these guys here move. Okay? There's one violation of that rule. That's, you can't see him well here. This is this thing that grows on top of this coral. It's called a sea squirt. And the sea squirt floats around the sea, and at some point it decides, I want to attach myself to this coral. And it sucks onto the coral. And the next thing it does, it starts suggesting its own brain. That's something that professors do. So, the idea here is, and this is an opinion, I don't have a proof, this is just a certain view of how things work, and that's my view and that of a whole set of colleagues of mine, and we're called the motor chauvinists. We believe the only reason why you have a brain is to move. And the only reason why you have vision is to decide where you want to move. And the only reason why you have memory is how you can decide about what happened in the past to make better decisions about movement. And of course, movement involves speech. I have to move my muscles to do that. And so effectively, for humans, the 99.99% of interaction comes from movement. The only other way the human brain can interact with the world is through sweat. That's the only other thing you can control that is not controlled by movement. Everything else is movement. Now, movement seems something very trivial because we can all do it at some point or another. But from an engineering perspective, it's actually not so trivial. So I'm going to show you now an example that's the result of a PhD thesis in Germany. So somebody spent 3 years of his life to do the following thing. He built a robot, and the robot task is to stack cups. Okay? So takes the cups, puts them on top, moves them. Perfect. Now, he would paste the cups again, and he wasn't too careful about placing the cups precisely. Oops. And the cups were not exactly stacked to the millimeter, and the robot fails. Now let's look at somebody else. who doesn't have a PhD in the topic. It's this boy here, and he does cup stacking, and this is a real-time video. He's the reigning world champion in cup stacking. Took him 6.5 seconds, and then he thought the bell didn't go off, so he's doing it again. Okay? That's how good we are at movement. And we're so good at it that we don't appreciate how hard it is for engineers to approach that. So learning to control to move is something we can't do. And there's several reasons for that. Because there's a lot of variability and a lot of infinite possibilities to do things. So for example, you can move quickly or you can move slowly. You can do it with many different joint configurations, hold the same position. You can do it with a lot of force and do it with little force. And so you can therefore catch the ball in many different ways. And this is a big, big problem for computers, because they have to explore all the possibilities in some way or another to figure out what to do. And we just do it. So therefore, to be a bit cheeky, this is Garry Kasparov, the last human world champion. And this is Deep Thought by IBM. And he beat this gentleman in chess. And since then, it's pointless to play chess. Computers can always beat you. So there we win out. This is an 8-year-old boy in Calcutta, manipulating a chess piece, more dexterous than the fanciest robots that we have. And the problem, just to stress that, is not that we cannot make fancy robot hands that have the mechanical degrees of freedom of the human hand. The problem is we don't know how to control them. We don't know how to program a computer so they can just grab something and manipulate it like this little boy. Of course, that's fancy and exciting if you're excited by these things, but there are objective reasons why I want to do that. So I want to understand how the brain works. But of course, all talk of movement disorders, but also aging, affect your ability to control movements. There are neurodegenerative diseases, Parkinson's, multiple sclerosis that affect your movements, and therefore we are interested in brain-machine interfacing. Understanding movements allows you to build better prosthetics, possibly smarter robotics, maybe think about better ways to do rehab, or build better computer user interfaces. So that's the motivation on a more objective scale, what we're doing, why we're doing what we're doing. And so, what we really are at is, we would like to be able to be as dexterous as Jamie Oliver as he prepares his Bayone. What we have at the moment, these are standard prescribed prosthetic hands, upper edge of the spectrum. What we really want to be able to do is to give a patient something like a robotic hand like this one here, and make him do what Jamie Oliver does. So when I started with this a few years back, I asked a simple question: what are the most important hand movements that are around there? And if you open a textbook, literally any textbook, you will find something like this. We have, this is from the Encyclopedia Britannica. Okay? We have the precision grip and the power grip or the power grasp. These are the two most important movements. So if you suffer a stroke and you're put in rehab, you would practice precision grips and power grasps. People will assess the scale of your disability, and based upon, for example, how much you're eligible to receive support, how good you're able to do these two things. Now, I usually don't believe what other people tell me until I've observed it myself. So with a PhD student of mine here, we put on something that we took from our research lab. This is a so-called cyber glove. So this is a wearable sensor that sits on your hand and measures all the movements of your hand. And we wore that thing for whole days in sequence. You come into you start your morning, you have breakfast, you put this on, you do whatever you do, and at the end of the day, you take it off and then go home. And we did that for a few days. And the first thing was, I didn't do a single precision grip the whole day. And I could live for that for 5 days without doing a single one. So clearly, this is not the most important movement, or one of the two most important movements. I just don't use it. And she didn't use it either, and many other students didn't use it either when we ran this through. And then it dawned on me that maybe a lot of things that we think we know about our movement behavior, we actually don't. And if you can trace that back through the literature, you end up in some textbook in 1730 by what we now call a neurologist, who said precision grips and power grasps are the two most important things we do. Without data, he just thought that was